Influenza, or flu infection, is a very common and highly infectious disease caused by the influenza virus. For some at-risk groups, flu can be very dangerous, with complications from infection responsible for tens of thousands of hospital stays and a significant number of deaths in the UK each year. Aging represents a key risk factor for worse outcome following respiratory infection, as shown here by the in-hospital mortality rates for influenza and COVID-19 infection. As you can see, with each decade of life, the mortality rate following infection increases. Vaccination programs targeting influenza have been extremely successful and are the best protection we have against an unpredictable virus. Several studies have shown that flu vaccination significantly reduces the rate of infection within vaccinated populations and reduces the severity of illness in a vaccinated individual that still gets sick. Influenza vaccines typically work by containing a harmless, inactivated form of the virus, which when delivered into an individual will generate a protective immune response against future infection. During aging, however, vaccine efficacy is reduced, leaving older people more susceptible to influenza infection and illness. This age-related decline is characterized by a quantitative decline in vaccine-induced antibody titers and a qualitative decline in terms of reduced antibody function and diversity of antibody-determining genes. In order to find ways to improve vaccine responses in older people, the first step will be to identify key contributing factors underpinning reduced generation of protective immunity. Upon vaccination, activated T cells provide help to B cells, which go on to differentiate into antibody-secreting cells. These antibodies are key to conferring protection against infection due to their ability to neutralize the virus. Vaccines are also important in generating memory B cells, which can generate a faster and more potent immune response in the case of future infection. Vaccine-induced antibody responses can arise via two main pathways. Antigen-activated B cells can differentiate into short-lived plasma cells that produce antibodies early in the immune response via the extrafollicular pathway. Alternatively, B cells can enter the B cell follicle, where they undergo class switch recombination before entering the germinal center response. The germinal center is a highly specialized microenvironment within secondary lymphoid tissues that can be subdivided into a dark and a light zone. Upon arrival into the germinal center, activated B cells undergo extensive proliferation in the dark zone, whereby they randomly mutate their B cell receptor genes in a process known as somatic hypermutation. B cells then test these rearrangements by collecting antigen presented by the follicular dendritic cell network in the light zone and interacting with specialized T follicular helper cells, where B cells that have successfully rearranged their receptor are positively selected and receive survival signals that allow them to either re-enter the dark zone for further proliferation and somatic hypermutation, or leave as long-lived plasma cells or memory B cells. However, it is increasingly appreciated that aging is associated with dysfunction and dysregulation within the germinal center response, including defects in T cell helper B cells, higher numbers of suppressive T follicular regulatory cells, and defects associated with FDC activation and expansion. In this paper, we aimed to investigate how the flu-specific B cell responses change following vaccination and aging, using single-cell RNA sequencing to provide an in-depth understanding of the phenotype, clonality, and germinal center selection of vaccine-reactive B cells in older persons. Influenza vaccines are formulated annually to combat antigenic drift and shift within circulating influenza strains, thereby generating the best immunity against the strains of flu that are expected to circulate in the coming season. In this paper, we studied a cohort of people vaccinated with a trivalent influenza vaccine within the 2016 to 2017 season. Two age groups of volunteers were selected, 10 individuals that were 22 to 36 years old, and 10 individuals between 67 and 86 years of age. Blood samples were collected immediately prior to vaccination, at day 7 and at day 42 after vaccination, and serum and peripheral blood mononuclear cells were collected to enable the analysis of antibody production and B cell subsets. All participants had H1N1 A California 09 neutralizing antibodies prior to vaccination, which were of a similar titer between age groups. We observed a significant increase in day 7 titers in the 22 to 36-year-old age group, which was sustained at day 42. In the 67 to 86-year-olds, there was a non-significant trend towards increasing titers of neutralizing antibody over the first week post-vaccination, consistent with previous studies. To investigate how the composition of the memory B cell compartment is affected by age, we index flow sorted hemagglutinin-specific B cells prior to and 42 days after seasonal influenza vaccination from all members of our cohort using an antigen-binding bait system. While both age groups increased the number of hemagglutinin-specific B cells after vaccination, the fold change after vaccination was larger in younger compared to older adults. Individual hemagglutinin-binding B cells were sequenced using plate-based SmartSeq technology. After quality control steps, UMAP clustering analysis was used to investigate the subset composition of hemagglutinin-specific B cells. This identified 5 clusters of B cells, each that could be defined by a unique set of B cell-related genes. In particular, UMAP cluster 1 was characterized by the expression of FcRl5, FcRl3, CD11c, and CD86, indicative of a population of atypical memory B cells seen in chronic infection, autoimmunity, and aging, and similar to influenza vaccine-responding cells described in younger individuals. When stratified by age and time point following vaccination, 2 subsets of hemagglutinin-specific memory B cells expanded following vaccination, including FcRl5 atypical memory B cells. This increase in FcRl5 atypical memory B cells was not significant in older people. These data indicated that the aged immune system does not generate memory B cells upon vaccination to the same extent as in younger people. We then validated these data using an extended dataset of healthy adults immunized during the 2016 to 2017 season. Of this, 6 younger and 4 older individuals overlapped with the original single-cell RNA-seq participants. These data confirmed that hemagglutinin-specific FcRl5 atypical memory B cells expanded to a greater extent following vaccination in younger compared to older individuals, resulting in significantly fewer hemagglutinin-specific FcRl5 B cells by day 42 in older people. To further investigate the phenotype of hemagglutinin-specific B cells, we performed t-SNE analysis of hemagglutinin-binding B cells at all sample time points in both younger and older donors from our extended cohort. 2 clusters were defined by high expression of FcRl5 and T-bet, indicative of atypical memory B cells, and showed temporal differences across the response to vaccination. These segregated into a CXCR3 high, CD11c intermediate, CD27 positive, and CD21 intermediate B cell cluster, defined as cluster 1, which transiently increased 7 days post-vaccination, and a CD11c high, CD27 negative, CD21 negative fraction, cluster 3, that peaked by day 42 post-vaccination. We postulate that these clusters may represent an earlier population derived via the extrafollicular pathway and a germinal center-derived population appearing much later in the immune response. Returning to our single-cell RNA sequencing data, we next aimed to investigate the rate of somatic hypermutation and selection occurring within hemagglutinin-specific B cell subsets. We focused our analysis on the 2 populations with the highest rate of class switch recombination, namely FcRl5 and Marks-positive memory B cells, with comparison to naive B cells as a control. Using the replacement to silent ratio as a readout of positively selected mutations, we compared selection rates in both the framework and complementarity determining regions of the B cell receptors across the 2 age groups. While the replacement to silent ratio within the framework regions was unchanged between age groups, there was a decrease in the replacement to silent ratio within the CDRs of FcRl5 positive B cells from older compared to younger individuals. This indicated that positive selection of this B cell subset after vaccination was impaired in aging. Furthermore, these data suggested a potential germinal center origin of FcRl5 positive atypical memory B cells following vaccination. Finally, we went on to investigate the potential germinal center origin of FcRl5 positive memory B cells using a publicly available dataset from Turner et al. In 2020. In this analysis, we compared single-cell transcriptomic data of B cells isolated from serial lymph node fine needle aspirates and paired blood samples taken at various time points post-quadrivalent influenza vaccination. B-cell receptor sequences of vaccine-reactive germinal center B cells identified within the B-cell compartment of the lymph node were used to identify daughter B cells with shared BCL clones that could be found within matched blood samples. These cells represented germinal center immigrants that could be identified in the circulation following vaccination. We found that at day post-12 vaccination, circulating cells clonally related to the germinal center were predominantly of an FcRL5 memory B cell phenotype, with over 60% of all germinal center-derived circulating B cells having a memory B cell phenotype at day 28 post-vaccination. These FcRL5 atypical memory B cells had high levels of somatic copy mutation, supporting the germinal center as a source of atypical memory B cells following influenza vaccination. In conclusion, we show that the formation of memory B cells is attenuated in individuals aged 67 to 86 years old. The formation of FcRL5 positive B cells that transcriptionally resemble TBET positive atypical memory B cells was particularly impacted. Using publicly available paired PBMC and FNA single-cell RNA sequencing data, we propose that a portion of FcRL5 positive B cells resemble circulating progeny from the germinal center reaction. Together, our data show that atypical memory B cells expand to a lesser degree in older persons and show reduced evidence of positive selection, indicative of an impaired germinal center response following vaccination in older people.